Okay, thank you very much for a kind introduction, and also thank you for the Molecular Frontiers organizers, supporters, and students, young people to attend this Molecular Frontiers Symposium. Now, I'm going to talk about this title, and you may wonder, snail? It's really talking about snail? And yes, this is the right-hand snail, Coiling snail. This is the left-hand Coiling snail, all right? We call it dextral and sinistral. Most snails, I'm working not land snail, but aquatic snail. And this one is I picked up in Japan, the sea, and these are dextral only snails. And this one is sinistral only snails called Physacuta. I'm going to talk about this a little bit later. This is a common pond snail. But always, exception is more interesting. And the snail I'm working on is Limnea stagnaris. It's a pond snail, and it has 98% dextral and 2% sinistral in the wild. And snail coiling is determined genetically. So I'm going to tell you a little bit about my background because you don't know anything about chirality, chiromorphology. Actually, chiromorphology is the word I coined myself from the two words, chiral and morphology. The concept is linking macroscopic and microscopic phenomena through chirality. Now you can see the very strange thing. This is the spectrophotometers called the CD, circular darkism spectrophotometers, that can measure solid state CD, and it has at least 2 photomultipliers at 2 rock amplifiers. It's very strange, but I've been working on this. So this is the chiral spectroscopy, and also I'm interested how molecules are packed in crystals. So this is crystallography. But I'm going to talk about this developmental biology. Well, I'm trained as a crystallographer and started doing spectroscopy for my PhD. Now I'm doing this for my career, maybe some other case I may talk about, but I'm not going to skip that one today. But you may wonder, what is chirality? You never heard about this. So chirality, this is a two-dimensional case, the scientist, if you make a mirror image of this, no difference, but all other letters are reversed. Look at the symbol of HKUST. That's complete mirror image, isn't it? So there's no chirality. But in fact, chirality is defined in three-dimensional world. And if the object cannot be superimposed on top of its mirror image, just like a hand, right hand becomes left hand, they are the different things, so that is the representative of chirality. Hand, foot, shoe, and electromagnetic wave and molecules. Of course, the molecules, S amino acid, this is the L amino acid, the bottom D amino acid. Of course, we use D, L, R, S to describe is this right-handed or left-handed. D, dextral, levo, rectus, sinistral, this is a Latin word, just means right and left. Plus and minus is actually due to the interaction of electromagnetic wave. And of course, there are achiral, many achiral ones. SOC, outlook our body, electromagnetic wave and molecules. Then you wonder, what is the origin of the word chirality? Where does it come? It's from chiro. That is the ancient Greek word that means hand. You know, if you know English, it's chiromancy, chiropodist, that's a chira, that's a hand. Who defined this? William Thomson in 1884. Do you know him? Yes, you do. It's the same one, Lord Kelvin. That's the absolute temperature's unit is K. Okay, so when he was knighted, instead of he becoming Lord Thomson, he becomes Lord Kelvin. This is the statue of Kelvin in Glasgow University. Why? Kelvin. Yes, this is in Kelvin Park, and River Kelvin flows into Glasgow University. So Okay, and chirality, that's become familiar to you now. Then you may wonder, all right, I know what the chirality means, but why it's so important? Why does it matter? But it is very, very important because biological world is homochiral at the molecular level. What does it mean, homochiral? It's described here. All living organisms on Earth use molecules of unique invariant handedness. In DNA, okay, so this is the double helical DNA. If you make it bigger, this is the base pairs, and this is the sugar phosphate backbone, and it uses only D-deoxyribose, no L-deoxyribose. It is true for RNA, it uses only D-ribose, not L-ribose. How about proteins? It is amino acid, and proteins, almost all, use D, no L, amino acid, no D. So that means biological world is homochiral, the same chirality. To understand how important it is, I made shoes and socks principles. Just like thermodynamic laws, I made 3. Principle 1, if you put your left foot into a right shoe or right foot into a left shoe, you will realize it immediately. But no difference in the case of socks. It's just like you shake hands. If both holds right hand, you can shake hands, but if other one holds the opposite hand, you cannot shake hands. The same thing. Principle 2, if a toddler wears father's shoes for fun, he or she cannot feel that left-right difference because it's far too big. Principle 3, a sock takes the shape of either left or right foot when worn. That's so, isn't it? And unless it's really loose. If worn for a long time, and if you don't wash it carefully, the part of the toe gets thinner. Now, if I say that, people will laugh, so I made science version of this, same principles. Principle 1, chiral compounds display chirality only when they interact with chiral compounds or chiral electromagnetic wave. If they interact with non-chiral objects, there is no chiral discrimination, just ear socks, okay? Principle 2, if order of magnitude is different, there is no chiral discrimination, even chiral objects interact. That means the dimensions, what sort of dimensions you are dealing with, or what sort of wavelengths, electromagnetic wave, you are dealing with. So that's important. Principle 3, dynamic chirality can be introduced to non-chiral objects when they interact with chiral ones. Dynamic chirality can be converted to permanent chirality. Okay, the socks, you can make a hole, that is it. So this is shoe and socks principle I made for easy to understand. So the consequence of biological world is homochiral is due to shoe and socks principle 1 and 2. It's important in the pharmaceutical and agriculture and food industries. This is not mirror image one, this is a diasterma we call it, but if it's aspartame, if you change the one to make mirror image, the L-phenylalanine to D-phenylalanine, it becomes bitter, although this should be sweetener, but it becomes bitter. This is the mirror image ones, and the fragrance is different. One is caraway seed, one is spearmint. Because our site, bad site of fragrance, detected site is chiral, and it's at the molecular level, they detect the difference, like our foot and shoe. And the more serious case is the thalidomide case. So drugs effect, one is teratogenic, one it works as sort of sedative. So FDA made new rules, the drugs have to be separated for the left and right, or it has to prove there's no harm if you give it racemate, that is a one-to-one mixture of left and right. So ordinary chemistry produces racemates, that means equal mixture of left and right. And what's happened is why we have only the deoxyribose and amino acid creatures. There are many things, but I don't think I'm going to spend too much time on it. But it's really exciting, but it's very difficult to prove, of course. So, go to my own research. So, chiromorphology, and this is sort of a schematic representation of my work. So, this vertical axis is the order of magnitudes in logarithmic scales. So, 2 is 100 meters, and 0 is 1 meter, and minus 15, minus 10 is angstrom. On the left-hand side, I put biological world. On the right-hand side is material world, because they are overlapping. But just make it easy, I just put it in separately. Now, so from the top, you can see the actual living organisms. Yeah. And going down to the inside organs and cells and proteins, DNAs, etc. So what I say is living organism, organs, embryos, proteins, genes, and here's molecules, and how these macroscopic things create these macroscopic chiral object is something I want to understand. So, linking macroscopic and microscopic phenomena through chirality to understand the mechanism. First, very quickly for this, and just want to do is the crystallization, because they have to recognize molecular chirality to form big crystals, or solid grinding, or transformation in solid state. And this is photoreaction, just like make dynamic chirality to permanent chirality. We can do this by reaction. And also the measuring using special spectrophotometers, which I developed. I had a patent on it, and it's quite interesting. Oh, this is just, I give only one or two examples only. This is the famous Pasteur's experiment, sodium ammonium tartrate. And why? D and L tartrate form only L get together to form crystals, or R to get only the crystals, or you can have one-to-one mixture. The difference is temperature, 27 degrees below or above, and this is Pasteur, and this is Scotti in Italy. There's no air conditioning in those days, so he got these racemic crystals. So I worked on it, how they recognize it a long time ago, before you were born, perhaps. I published a paper in 1981. Now, the another one is the spectroscopy. This is how you can follow the reaction in the solid state, or how the porphyrins actually aggregates in the solid state by the droplet of amine, or this is how this hornet silk will change the conformation in the solid state. This is just a film, but just 5 seconds exposed to steam, it starts changing the conformation from alpha helix to coil to coil to coil to cross beta. And the quick one, because Nancy is specialized in Alzheimer's disease, so I couldn't resist to put this one. And you just put the droplets of this, and it is alpha helix. But a while, if it gets dried, it changes very quickly in a very short spread of time to beta sheet. And you can measure this with my spectrophotometers. Now, I'm going to snail work. Okay, so most animals show bilateral symmetry, that means no chirality. But exception is always interesting, and this is my snail. But although we are bilateral, our internal organ is completely achiral. Heart is on the left-hand side, one liver, and gut goes one direction, and that is determined genetically. So how is it determined? Just And that is a molecule. And this is the case in mice. You see, it's at 7.5 day of embryo, there's a nodal flow. And at 8.5 days, there's a nodal, this is the name of the gene, that is expressed, and also PTX. I'll talk about this later. And that determines the heart is created on the left-hand side of mice. And there's very interesting work done in a Japanese group, but they are still debating. They are carrying some compounds or the stimulus of physical flow, but it's very interesting work. Now, I'm working with snails, and you wonder why snails and why in particular Limnaea stagnalis. Because both dextral and sinistral species are found in the world, as I talked to you earlier. Dexterity is dominant. Then also the symmetry is displayed outside as well. You don't have to cut open to see whether it is left and right. In human beings, 1 in 10,000 has situs inversus, completely mirror image internal organs, but you won't see it. Okay? Hermaphrodite. That means there's no male and female difference. And it both self-cross and cross. That is easy to do some genetic work. And snail huntington's depends on the direction of spiral cleavage of embryo. That was reported in 1894, that early, just like this. That was reported like this. If we go into the sinistral snail, it's from 2 cell to 4 cell to 4 to 8 cell stage, it rotates anti-clockwise way. But in the case of dextral snail, it rotates clockwise. It's so clear-cut that has been reported. And body asymmetry follows a maternal mode of inheritance. So not your own gene, but your mother's, okay? So if your mother right-handed, you're born with the shape of right-handed. That is, and also it says determined by a single gene. But this is reported in 1923 to 30 by Stuyvesant, Boycott, and Diver. That's long before 1953, the DNA was discovered, and this inheritance matter. So in a sense, this is a Mendelian gene, but there's something important in it. That's the reason I started working on it. So it's just like this, dextral and the sinistral snails. And the first discovery is dominant dextral and recessive sinistral embryos are not mirror images of each other at the crucial chirality determining step. That is a third cleavage from 4 cell to 8 cell stage. So it is like this. Okay, so the top is the sinistral, bottom is the dextral. So look at that video. Yes, it rotates, and that rotates. So this is really massive talk it shows, but this is what it's actually doing. But if you look carefully, symmetry is broken. Here on the one, at this phase, metaphase, anaphase, the cell is already deformed in spiral direction. I named this SD, spiral deformation, and also spindles helically twisted. So I call this SI, spindle inclination. So small micromeres are inevitably produced, and you get this one. On the other hand, the sinistral snails, these are radially placed, and there's no chirality, and the small one, micromeres produced just above the big 4 macromeres, and when it cleaves off, it rotates. So symmetry is broken. Then, I realized actin is important because when you inhibit the actin polymerization, there's no membranes formed, but spindle is not tilted. So spindle is not pushing the shape of the cell, but actually, if you look at this side, if it inhibits the microtubule polymerization, there's no spindles, but there's a substantial spiral deformation. So we got is actin must be important. Now, in many textbooks, even now, it shows this figure. So they are completely mirror image all the way through, but I hope it will change it like this. At this important part, mirror symmetry is broken. But if you use Pfizer-Acuta sinistral-only snail, then you will see a mirror image relationship, but it's a different snail. Okay, so if it's a dominant, its mirror symmetry relationship holds, but within a species, it's not. And it's coming out in current biology in top, in the figure. And it's quite a nice thing it's written here is the results of an exciting and elegant new study diffuse this model, and it's quite a nice thing showing that right doesn't have to be the mirror image of left. I thought it's quite a nice way to put it. Anyway, then I want to prove it's really a single gene locus. So it takes ages to do this work, but if you cross sinistral and dextral snails, pure sinistral, pure dextral snail, it's very difficult to cross because sexual organs has a chirality, but you can manage to do this in this snail. And you can produce F1. F1 becomes always inherit the dominant gene, right-handed determining gene. Then you pick up this to cross with the sinistral one, and the sinistral one make it mother. Then it produces F2, and if it's inherit the dexterity determining gene, of course it all has sinistral shape because mother is sinistral. But you have to look at the next generation, and if it's inherited, it produces right-handed offspring, okay? And if it's not inherited, it produces left-handed offspring. So you wait to see what offspring it produces, and you cross this with pure sinister mother. And that is produce offspring. So it's quite a long way to go. For example, if you come to F6 stage, right-handed genome becomes 1.6%, and sinister snail genome is 98.4% on average. So that is quite nice, and if you keep going, you get F10 congenic snail. Dexter, 0.1%, sinistral, 99.9%. And if inherit that dexterity determining gene, it's always produce SD, SI, rotates clockwise way, and it produces dexter snail. But if it's not inherited, it is no SD, no SI, and becomes sinistral snail. So it must be single gene locus. That's what we realize. Then we want to identify what is the gene. So now I'm not going to bore you, but this is 7 years of tears and perspiration, background and linkage markers. And, I'm just telling how easy, nicely, but no, lots of failures behind. But anyway, out of 15 candidate genes and 13 is maternal, out of that, we picked up the actin-related genes, and we did whole sequence. We could look at this. So unfortunately, there's a quite long code spot for the combination, so we have to deal with this 700 kilobase. So we did the sequence, and only this LS-DR1, LS means Limnia stagnalis, DR means diaphanous gene. The DR1, only that one has a difference between the sinistral snail and dextral snails in the wild. But there's another one called LSDR2, it's randomly repeated. What's the difference? This one, it may not be that easy to see, but it's AACCTC. This is the dextral one. The sinistral one, AACTCA. 1C is missing. That is the only difference we could detect. What is the consequence? If that is missing, after a while it becomes stop codon. So that protein is not produced. So sinistral snail has only LSDR2. On the other hand, dextral snails have this 1 and 2 both. But surprisingly, LSTR1, LSTR2 has 89.4% homology. Isn't it amazing? It's so similar, but this is actually the results we obtained. So, tandemly repeated LSD1, LSD2 gene exists for the right-handed, but sinistral one lacking LSD1. And we made antibody. Yes, we had the importance of antibody this morning. It's sometimes very difficult to make. But yes, if you look at this, LSD1 has no, this Western blot we call it, no proteins for the sinistral one, and for the dextral one, we're just following the development. So up to this late blaster stage, it exists. Of course, this is before the zygotic, I think, expression starts. So this is quite interesting. But this is a candidate gene. There may be some other genes involved, and we may be missing that. We're not observing it. So we have to prove if that is the single gene that determines chirality. I thought I should do the CRISPR-Cas9 knockout, not knockdown, knockout. So this is where the wild type mutation is here. But because 89.4% homology, I could not set the guide RNA there. Therefore, only the place I could do is FH1, forming homology domain. Then I did it. So I'm going to make the short story. So inject it, and F1 is mosaic, and F0 is mosaic, F1, and it becomes homogeneous. So we check what children, what offspring F1 produces, and it's surprisingly clear-cut. Okay, knockout LSTR1 gene, only that one, it's totally dextral genetic backgrounds, completely pure right-handed snail, and knockout one gene, LSTR1, by CRISPR-Cas9 genome editing. Now, look, this is the genotype F1. If you look at it, this is the red one is right-handed, and blue one is left-handed. If the both alleles are non-frame shift mutation or wild type, it always produces dextral snail. But if, like here, del3, del10, because right-handed, del3, no frame shift mutation, del4, del10, of course, both are frame shift mutation, so it produces left-hand snail. So just look at it. If LSTR1 is knocked out and LSTR2 only, it becomes right-handed snail. And it goes to next generation. Now it's F17 generation. So no exception, it keeps going to produce sinistral snails. Otherwise, completely dextral one. And also the anti-clockwise rotation, clockwise rotation, and also SDSI also matches with this. And also you check the antibody, then again, it's very, really clear-cut, you know. Del3, del5, del5, you don't have this protein, but if it's del3, 10, or wild type del5, yes, it shows, so it's very clear-cut. And also we realized you don't have to wait until the third cleavage that becomes 4 to 8 cell stage. At the first cleavage, so at the fertilized stage, there's a chirality already determined. So if you look at the first cleavage, that means fertilized eggs become 2 cell stage, you can observe rotation, and again, clockwise, anti-clockwise completely match with the genotype. So it is really fascinating. And also, nodal PTX, I said I'm going to talk about this. The nodal is really matches with the genotype. If you look at this, this is the wild type. Sinistral one, L is on this side. And wild type dextral is mirror image site. But if you do this CRISPR-Cas9, del 5, del 5, it goes to sinistral type, and del 7 plus ins 1, del 5, that's not, one is not frame shift, it becomes just like the dextral snails. It was very, very clear cut. So we could create just, generation to generation, the right-handed background snail to left-hand snail by knocking out one gene. So we can say that is actually the hand is determined. And we checked another snails, and this is the Physacuta, sinistral only, has only the type 2 one as well. But this is with tampered gene, but then more another exciting thing is the mechanogenetics. I'm not doing anything to the gene, but just manipulate it by hands under the microscope to change the orientation of micromeres and macromeres. So that's like this. It's SDSI, so dextral one, chirality develops early, so start pushing up to here. For the sinistral one, chirality develops a bit later, so we started from here and push it here. Then we culture these things. That is the most difficult part. But we managed to create animals, snails, and surprisingly, it's completely opposite, This is the sinistralized dextral snail. It's the dextral snail, but we push it under the microscope at embryonic stage. It becomes sinistral snail. And this is the sinistral snail, but if it's manipulated under the microscope to put it the dextral way, its orientation just becomes dextral snails. That is really amazing. And the positional nodal, it's also converted to the mirror image site. That is the reason we can obtain really proper snails, and it produces offspring that detected or dictated by the gene. Then you may say, oh, that's dimorphic. You worked on the exception. So it may not work for other snails, so it worked on Physacuta. That's a sinistral only snail. Can we do the same thing? Yes. We created dextral snail, which no one ever seen it. But of course, next generation goes back to the gene it has, so it goes back to sinistral, but we could create the dextral Physacuta. That means the mechanism of making the body is common. So this is sort of a summary, so up to here. So you can go back to the original, and when it up to that is intramolecular, here to come intermolecular communication of information on chirality. This actually appeared about 1 month or 2 months ago in annual reviews on Cell and Developmental Biology. The single author had to work hard for writing this, but anyway, I hope you can read it. Then you wonder, what is the role of this DR1, DR2? What is doing? Actually, this is the very fundamental gene we all have. It's ubiquitous in all eukaryotes have, and it's involved in actin filament elongation. And this is the sort of auto-inhibited one. It forms dimer, and it is an auto-inhibited one, and interaction role, etc., it becomes this active form, and this porphyrin actin monomer is get together to get nice actin filament. Now, in the wild, there's a mutation here. So these green parts, there are many domains in this protein. Green part is missing. So we know that is an important part. However, now I did CRISPR-Cas9, and only these yellow domains exist. That means this N-terminal part is not important in determining the handedness. Now try to work out, what is doing this. And of course, it's difficult, but I'm working on that. So trying to understand how the gene determines chirality. from one cell stage to an individual organism through biological hierarchies using biophysics, molecular and developmental biology. I was trained as a physical chemist, so I tried to make my advantage. And so here, this is a cytosine versus 1 in 10,000. Human beings has a complete mirror image internal organs. In the case of mice, I told you 7.5 days, there's a nodal flow, 8.25 day nodal is expressed. Look at the case. 12 hours after the first cleavage, nodal is expressed. So, and also it's determined at the one cell stage already. And at this stage, third cleavage, you cannot change chirality anymore. Before, by micromanipulation, you can change the chirality. So it is really interesting. So it's appeared, this is actually from cover of Current Biology. And this actually in Nature, it doesn't appear, just I took it from inside. This is the front cover of Development. They're all different. This is the wild type, left and right. This one, I manipulated. So this right-handed one is created from left-handed snail. At the embryonic stage, I pushed it. And this sinistral one is created from dextral one by micromanipulation. And this one, the right-handed one is a wild type one, and this is the one created by knocking out LCD1 gene, which changed to sinistral one. Okay, now I have to be quick. So I'm working on this, but I decided to apply this technique because I'm still only the one successful in genome editing for the mollusks. So I'm working in two. So one is control of schistosomiasis. Because the intermediate host is the freshwater snail, Biomphallaria gravulata, that is phylogenetically close to Limnius stagnaris. I worked on dextral and sinistral one. The second one is the brain science, just the memory, and Alzheimer's disease or Parkinson's disease or Huntington's disease, etc. You may be wondering why. So I just quickly, last couple of minutes, I'm going to explain that. The controlling the schistosomiasis, this is a snail-borne neglected tropical disease assigned by WHO. And about 250 million people worldwide is suffering, and 200,000 people die each year. Now, snail, called Biomphallaria gravulata, is the specific intermediate host of the parasite, and inside the body of this snail, it does a metamorphosis. So from Miracidia, it comes to sporocyst first, sporocyst second, it changes to cercaria, and cercaria gets into humans, just going through skin. So if you put the foot in fresh water, if this lives, you had it. So it's an incredible problem. But in the wild, there is some snail line which is resistant. It doesn't produce cercaria. So somehow, this Miracidia goes inside the snail body, but it seems dead inside. So try to work out the immune system of snail to save these peoples. So I'm working on it, and I got a grant, NIH, and actually, it's too early to stage. I'm just writing a paper. I succeeded to create knockout gene, and it's second generation. So, but it's not that simple. So you have to wait for that. For another one, this is Alzheimer's disease, and you may be worried, but this has quite good long-term memory, and there's no blood-brain barrier. So if you apply A-beta, for example, near the foot, it goes into brain very quickly. And we are doing this taste aversion experiment. Sweet, bitter, sweet, bitter. Then the cleverest one I have is remembered for 57 days. So I try to work on, because the neuron is, the number is very small, rather big neurons, and also it's surprisingly asymmetric. So I have to say sinistral and dextral snail, which is more clever. So it's almost a joke. So I'm working on this. So these are the acknowledgement. These are the people have been working for a long time ago now, but for the CRISPR class and first paper. And I don't have really big groups, so I'm not allowed to have students, but some students come and join. So I thank the University of Tokyo. I worked for a long time in Tokyo University of Science, and now currently I'm at Chubu University. And do you know the Carolian, the Lewis Carroll's famous book, Through the Looking Glass, What Alice Find There? This is actually 150th anniversary, 2021. They had a special issue. This is a really shining cover, like a mirror. And I'm honored to write one chapter in this book. And this is my snail. So curious to look into the mirror. So I hope you are just as curious like Alice or this snail. Oh, what a wonderful word about chirality is. Okay, thank you for your kind attention.